Hello there, everyone. How are you all doing? I hope you're all doing well, and I hope you're going to enjoy this lesson. I hope you're ready to learn. Welcome back to another cellular biology lesson. We're still looking at the cellular organelles of the eukaryotic cell. In the previous video, we obviously look into the nucleus. We look into its components like the nucleoplasm, the nucleus, and the nuclear envelope, and we have studied the processes that occurs within the nucleus and how it contributes not only to the functioning of the nucleus but also to the overall development of the eukaryotic cell. So now, in today's lesson specifically, we're going to study the ribosomes, which is going to be obviously interesting. It's going to be intriguing. So what are ribosomes, including its structure and functions? Ribosomes are minuscule or tiny but essential organelles that synthesize proteins by utilizing amino acids through translation, which involves translating the genetic information that is stored and encoded in the messenger ribonucleic acid, also known as the mRNA, as the genetic information contains the instructions in order to assemble the amino acids together to form a polypeptide chain and thus a synthesized protein as a result of protein synthesis, and the synthesized protein will be utilized for various key cellular processes which contributes significantly to the overall organism at the cellular level. In eukaryotic cells specifically, the ribosomes are produced from the nucleus, which is an area of dense chromatin within the nucleus, and the ribosomes are then distributed throughout the eukaryotic cell, in which some of the ribosomes are attached to the rough endoplasmic reticulum, in which the rough endoplasmic reticulum is a type of endoplasmic reticulum that produces proteins as it contains ribosomes, the tiny organelles that involves the synthesis of proteins through the utilization of amino acids. Eukaryotic ribosomes are known scientifically as ADS ribosomes because their sedimentation coefficient is ADS, in which eukaryotic ribosomes are composed of two subunits: a large subunit, which has the sedimentation coefficient of 60S, and a small subunit, which has the sedimentation coefficient of 40S. During protein synthesis, these two subunits come together to create the entire ribosome, which has an ADS sedimentation coefficient. So just to clarify what is sedimentation coefficient, sedimentation coefficient is a measure of the rate at which particles or cellular components sediment during ultracentrifugation. The S unit is derived from the Zithberg unit, if that's how you pronounce it. Forgive me if I didn't pronounce it correctly. Anyways, the Zithberg unit represents the sedimentation coefficient of that particular cellular molecule or organelle, like the ribosome. Furthermore, it indicates the pace of sedimentation rather than the size in a straightforward manner. And as you can see below, here's the equation of the sedimentation coefficient. Yeah, it looks very complex at this stage, but with practice you can obviously understand, but we're not looking to that for now. Then finally, we'll look into studying the translation. Translation is obviously essential for the ribosomes, as without translation, there'll be no protein synthesis, and hence ribosomes will be useless, practically and theoretically useless. Ribosomes strives to obviously synthesize proteins for the utilization of amino acids, and that can only be done through the nucleic acids, particularly the mRNA and tRNA. And these are obviously part of the translation process, and the translation process initiates because of these nucleic acids that are produced and derived from the nucleus. So obviously we want to look into the translation and study it thoroughly, and we want to have an advanced understanding of translation. So what is translation? Translation is an essential biochemical and cellular process in which a cell creates proteins by utilizing the genetic material that is stored and encoded in the messenger ribonucleic acid, also known as the mRNA. Translation involves translating the genetic information that is stored within the messenger ribonucleic acid, as the genetic information contains the instructions in order to assemble the amino acids together to form a polypeptide chain and thus a synthesized protein as a result of protein synthesis, and the synthesized functioning protein will be utilized for various key cellular processes. The initial step of translation is tRNA charging, in which the transferribonucleic acid, also known as the tRNA, plays a crucial role throughout translation by carrying specific amino acids to the ribosome, where these specific amino acids are incorporated into the growing polypeptide chain. Secondly, the next step is initiation, which marks the beginning of translation as the ribosome begins to translate the genetic information that is carried and encoded in the messenger ribonucleic acid, as the genetic information contains the instructions. Then the third step is elongation, in which it is during which where amino acids are added to the polypeptide chain, in which elongation is where amino acids are added to the polypeptide chain, and thus the polypeptide chain gets elongated or essentially extends. Then the final step of translation is termination, in which termination signals the end of protein synthesis and the release of the completed synthesized protein, which will be obviously utilized for various key cellular functions, such as muscle growth, tissue, and repair, beginning at the cellular level. So yeah, that pretty much wraps up the whole second lesson, the second eukaryotic cellular biology lesson on the ribosome and the translation process. And obviously we got the images here for reference because obviously there will be visual learners, and it's obviously a great way to understand these components and functions with these images, and you can see how these interactions occur uh, in a visual sense. But yeah, that pretty much wraps up for this lesson. Thank you very much for watching and paying attention, and I hope you learn and keep up your studies in biochemistry, microbiology, biomedical science, and whatever biological field you're into. Thank you.